This is Libby speaking. In conservative replication, all old DNA ends up in one daughter cell and all new DNA ends up in the other daughter cell. If the Mendelssohn-Stahl experiment were to display conservative replication, the E. coli grown in the medium would still contain nitrogen-15 at generation 0. When the first replication cycle ends, we would be left with one daughter cell containing all nitrogen-15, which is the old DNA, and one containing all nitrogen-14, which is the new DNA. This pattern would continue throughout the replication cycles, with the nitrogen-15 cell producing one nitrogen-15 daughter cell and one nitrogen-14, and the nitrogen-14 cell producing two nitrogen-14 daughter cells. So we are left with the nitrogen-14 cells outnumbering the nitrogen-15 cells in the long run.
DNA model, the density pattern would look like this. If the conservative model was true, the heavy N15 DNA would never actually disappear. The bar representing heavy DNA would continue to get smaller and smaller. Since the actual data from the first generation had one bar of hybrid weight DNA, rather than two bars of separate N14 and N15 DNA, the conservative model is shown to be inaccurate after one generation.
Replication. The original DNA double helix breaks apart into fragments. Two DNA molecules that are mixtures or hybrids are produced as a result. Each individual replicated strand is a combination of both parent and daughter DNA in varying amounts. The patchwork DNA would also contain a larger fraction of nitrogen-14 with each successive generation, and it gradually decreases the density of the DNA over time.
This is Sabrina speaking. There were two bands when the second generation DNA was centrifuged, with one being at the same place as the first generation intermediate bands and the other being higher, labeled as nitrogen-14. Because there were distinct bands at the positions of a hybrid molecule and a light molecule, dispersive replication is ruled out because if it had occurred, then a light molecule would not have been possible to get, since molecules would have to have a mix of old DNA and new DNA. There would be a mix of molecules with light and heavy sections because of this, and more of the lighter nitrogen-14 as generations continue, meaning that there would have to be one band that gets lighter each generation.